Hello, my gentle and of course very modern apes. Stop me if you've heard this one. Humans and chimpanzees are 99% genetically identical to one another. Or maybe you've heard it like this: humans are 99% chimpanzee. This is a number that shows up all the time in textbooks and in museums and on YouTube videos to talk a little bit about the similarities between humans and chimpanzees, given we are one another's closest living relatives, to illustrate just how little separates the two of us as species. But how many of you out there know what that number actually means? What it's actually measuring? What it's actually comparing? How many of you out there know how this number has perhaps changed over time? How we actually compare species using comparative genomics, and perhaps where we stand on the similarities between humans and chimpanzees now that a new paper with the complete genomes of several great ape species and one lesser ape species have come to light via a new Nature publication. Today, I want to explain to you how similar humans are to chimpanzees using the latest data available, and I want to explain that a little bit through the history of how we came to understand just how closely related we are to our closest living relatives. I want to explain to you that there are many ways to compare genomes, but that no matter how you do it, humans and chimps still hash out as one another's closest living relatives, as more similar than rats are to mice. Are humans and chimpanzees 99% the same genetically? Why, yes, but also no. Let's get into it. Since humans have had the time to wonder about the natural world, we've wondered about our place in it. Anyone can look around and recognize that some pairs of animal species are more similar than other pairs of animal species. A dog is more physically similar to a wolf than it is to, say, a cat. One of the first naturalists in the West to try and make sense of the variety of life on the planet was Carolus Linnaeus, the father of taxonomy. In the 1700s, Linnaeus devised the classic taxonomic system we all know and love: kingdom, phylum, class, order, family, genus, species. And when he looked at the wide variety of critters that he shared the earth with, he immediately recognized that humans were more similar to one group than we were to any other. Humans had all of the same characteristics as other apes did, like orangutans, chimpanzees, gorillas, bonobos, and gibbons. And Linnaeus was not the first person to notice this. In the late 1600s, British anatomist Edward Tyson dissected a chimpanzee and noted that chimps actually shared more physically in their bodies, as well as specifically in their brains, with humans than they did with monkeys. This made the rest of the Western world very uncomfortable, because humans were supposed to be separate from the rest of the animal kingdom. So Linnaeus' solution was not to denigrate humans by pushing us down to the level of some other group of animals, but to elevate the other members of this specific group. The apes and humans became united in anthropomorpha. Critically, this was before the advent of the theory of evolution by Charles Darwin and Alfred Russel Wallace. So, similarities in physical form didn't necessarily mean that two organisms were related. In fact, in Linnaeus' time, most people held to a concept called species fixity. This is basically the idea that species don't, and in fact, can't change. All species were created as is, and similarities that you see are just similarities in design. So dogs and wolves aren't necessarily related, they just share a common template. All of this changed in the 1800s with a few major discoveries. First, the field of paleontology was born, and we realized that there existed species of animals in the past that share lots of similarities with species today, but which no longer inhabit the Earth. And the second was Wallace and Darwin's theory of evolution by natural selection. The idea that species can, and in fact do, change over time due to natural selection acting on variation within specific populations. This was sort of a duh moment for much of the scientific community because it so eloquently explained the natural world, everything from biogeography to the fossil record to the process of domestication. But one piece of the puzzle was missing, and that was how these characteristics, which seemingly link organisms in ancestor-descendant relationships, kind of worked, how these characters were passed on from parent to offspring. The discovery of genetics by Gregor Mendel, and then elaborated upon by many, many different people in the early 1900s, changed all of that. Before we go any further, I'm gonna have to give you a quick primer on genetics, so you buckle your seatbelts. We knew organisms were made up of cells, but it turned out that the secret to evolution lurked inside those cells in the form of DNA. DNA wasn't officially discovered until the middle of the 20th century, a molecule that contained the genetic instructions for the development and maintenance of every living organism on the planet. Everybody's seen DNA before. It's like a twisty ladder, but that twisty ladder is actually a little bit more complicated than it seems. The ladder of DNA has two sugar phosphate backbones which make up either side of the ladder, and the rungs of the ladder are nucleotide bases. There's 4 of them: adenine, thymine, guanine, and cytosine. And those guys always pair in consistent ways: adenine with thymine and guanine with cytosine. DNA can be crudely broken down into 2 categories: protein coding DNA and non-protein coding DNA. Protein coding DNA codes for proteins. It's stretches of DNA that code for a specific protein, in fact, and these stretches are called genes. Because genes do the heavy lifting functionally and structurally speaking, oftentimes protein coding regions are called functional regions of the DNA. Non-protein coding regions are generally considered non-functional in the sense that they either don't have any known function or they perform some regulatory function. Protein coding regions only make up a tiny portion of our genome, approximately 2%. The rest is either regulatory, telling genes when to turn on or off, or most of the time, it's just non-functional redundant DNA. Now, the DNA ladders that you see depicted in cartoon format are not, in reality, short. They're composed of billions of base pairs, with the complete set being known as your genome. Every single one of your cells, with a few exceptions, has a copy of the entire human genome located within its nucleus, so it has to be packaged efficiently. DNA is wound tightly around proteins called histones in this sort of pearls on a string diagram that you often see, and those fibers, the chromatin, are what form each of your chromosomes. To simplify it in an analogy, you can think of your entire genome as the book of you. And each chapter within that book is a chromosome. The paragraphs that make up those chapters are traits, physical characteristics, and the sentences that make up those paragraphs are genes. The words that make up the sentences are codons of amino acids, and the letters themselves are the base pairs. When your body needs more of a specific protein, it will open up the book of you and go to the sentence that codes for that protein. It will unzip the DNA at that specific portion and begin a process called protein synthesis. Because you're always making new proteins in specific parts of your body to keep you alive and running, it's important that every cell has a copy of the recipe book. The majority of the cells in your body contain 46 chromosomes, 23 from your mom and 23 from your dad. These are diploid. Your sex cells, on the other hand, are haploid. They only contain 23 chromosomes. And the purpose for this is because we're sexually reproducing organisms. When a sperm meets an egg, they combine their respective 23 pairs to create a new organism with a novel combination. That new novel organism is in part the result of sexual reproduction itself, because you have 2 haploid cells from 2 different parents creating a new novel combination, and in part because each of those haploid cells from each parent is itself slightly different from the parent itself due to a process in meiosis called recombination. So, you are an organism composed of over 1 trillion cells, and in the majority of those cells within the nucleus, you can find your genome, the entire instruction manual to make and maintain you. Because almost every one of your cells contains that entire human genome, when your cells replicate, they have to replicate the entirety of the genome within their nucleus in a process called DNA replication. DNA replication occurs in both mitosis, the replication of somatic cells, and meiosis, the replication of sex cells. The key difference is that at the end of mitosis, you have 2 diploid cells, and at the end of meiosis, you have 4 haploid cells. All of this sounds wonderful and intricate and complicated, and it is, but it's also the product of billions of years of evolution. which means it is painfully imperfect. When there is an error in DNA replication or in protein synthesis, we call that a mutation. And if that mutation happens in the sex cells during meiosis, it is passed on to the offspring. These mutations will contribute to the differences when you compare the full genome of a kid to the full genome of either of their parents. So you are a combination of a haploid sex cell which has undergone recombination from your mom and a haploid sex cell which has undergone recombination from your dad, both of which have potentially undergone mutations and then been combined to create you. This is why you are overwhelmingly the most similar to your parents, genetically speaking, but you're not identical to them. And this is what makes sexual reproduction so wonderful. It adds new variation through these processes to a population and makes them more robust in the face of change. And it also means that genomes can tell us how closely related two organisms are to one another, because similarities in genomes and changes in genomes are related directly to reproduction. It's sort of like the track changes function in Microsoft Word. If I sequence my genome, my sister's genome, my cousin's genome, and a stranger's genome, we'll see our known family tree recapitulated by our genomic similarity. Me and my sister will have the most similar genomes, and this is because we share a recent common ancestor in the form of our parents. Then the two of us will form a triad with our cousin. The three of us will have genomes that are more similar to each other than any of the three of us has, similarity-wise, with the stranger, because the three of us share a common ancestor in the form of our grandma. That stranger forms the outgroup. We know this for a fact because we have family trees that exist today made up of still living people whose genomes we can then sequence and compare to outgroups like strangers, and their genomic similarity will reflect their known family tree. This is non-controversial, which is why paternity tests hold up in court. Genomic similarity very simply reflects relatedness, and the more similar the full genomes are between two individuals, the more closely related they are. Across all humans, we see very little difference genetically speaking. The number that you've probably heard as a comparison to the 99% genetic similarity between humans and chimpanzees is that humans are 99.9% similar to one another. And this is the case with many species, where the members within that species are all more similar to one another than any member within that species is to a member outside of that species. We've done these types of comparisons with dog breeds, so all dog breeds are more closely related to one another, are more genomically similar to one another than any of them is to like a gray wolf. But you can add more species into the mix. For example, if you sequence the genome of 3 different dogs, a wolf, and a coyote, what you will find is that the dog genomes are more similar to the wolf genome than they are to the coyote genome. And this is for obvious reasons. We domesticated dogs from wolves. But like our analogy with siblings, cousins, and strangers, the wolf genome will also be more similar to the dog genome than it is to the coyote genome. So when you compare the similarities and differences of these genomes together, the result is a family tree where dogs and wolves are one another's closest living relatives and coyotes are the outgroup. If we add a fox into the mix, what we'll find is that dogs, wolves, and coyotes are all more similar to one another than any of them is to a fox. Add in a domestic cat, and you'll find that dogs, wolves, coyotes, and foxes are all more similar to one another genomically than any of them is to the cat. And you can do this with the entire tree of life. A family tree at the species level is called a phylogeny. What's cool is that phylogenetics more or less recapitulates the groupings that we created on the basis of general physical characteristics, on the basis of morphology. Like, people could more or less deduce that the fact that dogs and wolves have more physical characteristics shared between them means they're probably more closely related than either of them is to, say, a cat. But genetics effectively proves many of these relationships. Like I said, it's kind of like the track changes function in Microsoft Word. And it helps that this is supported by the fossil record too. Dogs are actually more closely related to bears than they are to cats, and we have dog bears in the fossil record. Cool. But one of the really big groups that genetics threw a massive monkey wrench into, is the similarity between humans and other apes. So like I said, we've known for a really long time that humans are most similar to other apes than we are to anything else. But the way that that family tree hashed out before the advent of genetics was very different. Humans were grouped in the superfamily Hominoidea, the apes generally, but then Hominoidea was split into 3 separate families: Hylobatidae, the gibbons or the lesser apes, Pangidae, the great apes, and Hominidae, the humans. That would mean that all of the other great apes, so chimpanzees, bonobos, gorillas, and orangutans, were more similar to one another than any of them was to a human. We were apes, if barely, the exceptional ones, the standouts. And this makes sense morphologically because, superficially, it does seem like chimps and bonobos share more with gorillas than they do with humans. The cracks in that notion formed first with immunological work that showed that when you compare humans, chimpanzees, gorillas, and orangutans are actually the odd man out. But the real first major blow came in 1975 when Mary Claire King and Alan Wilson published Evolution at 2 Levels in Humans and Chimpanzees and revealed that chimps and humans are 99% similar when comparing large sets of proteins between the 2 species. This did 2 things right away. First, it cemented chimpanzees as the closest living relatives to humans, and 2, it revealed that physical differences maybe don't track as tightly as we thought to protein coding region differences. Maybe the real differences between humans and chimpanzees lurked in that non-coding DNA, and for that we would need a more completely sequenced genome. The human genome was sequenced in 2002 as a part of the Human Genome Project, but put an asterisk there, and the chimpanzee genome was sequenced just a few years later. And with the 2 of them in a decently complete state, we were able to compare them and get an idea for the percent similarity when looking at the whole thing, instead of just the protein coding regions. Humans and chimpanzees hashed out as about 96% similar when looking at the entire genome and taking into account single nucleotide polymorphisms or 1 base pair changes, as well as large-scale insertions or large-scale deletions. The number was about 99% when you took insertions and deletions out and just looked at the genome with regard to those single nucleotide polymorphisms. And of course, protein coding regions were even higher than that. Now I know what you're thinking. What's an indel? What's a single nucleotide polymorphism? Put a pin in it, we're going to talk about it in a second. It's worth noting here that the protein coding percent similarity between humans and chimps is quite a bit higher, I would say, than the whole genome percent similarity, whether we're just looking at SNPs being included or also including indels in addition. Now remember, the protein coding portions of the genome are not very extensive. It's about 2% of the genome that's protein coding and 98% that is non-coding DNA. So for 2% of the genome, humans and chimpanzees were more than 99% similar. That's not very much of the genome, but we have to remember it's also arguably the most important part of the genome. It's the part of the genome that does stuff. Whereas for the rest of the genome, depending on whether you're including SNPs or SNPs and indels, humans and chimps were somewhere in between 96 and 98.8% similar. So how similar are humans and chimpanzees? Over 99% for protein coding regions, and somewhere in between 96 and 99% for the entire genome. You would think that that would settle it, but not quite. There were still a lot of issues and a whole lot of questions surrounding the question, how similar are humans and chimpanzees? What did 99% and 96% actually mean? How did it compare to other pairs of organisms? How would it compare as we continued to sequence the genomes of other mammals, of other primates, and of other apes? Like, is 99% for protein coding regions high? Is 96 for the whole genome high? Or are they low? We needed some context. In addition, that 96% came with some caveats about the way the 2 genomes were compared. For example, how did the comparison take into account different types of mutations? How did it take into account differences in genome size? More on that later. But the biggest issue, arguably, of these 2 comparisons, and this is no fault of the authors, this was very clear in the literature itself, it's just something we have to note, is that the genomes weren't actually completely sequenced. Remember, the human genome is billions of base pairs long, and so is the chimpanzee genome. And across that genome, there are many sections that are just kind of a mess. It's thousands upon thousands upon thousands of base pair long sequences that are just repeats, ATA, 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 over and over and over again. There were large chunks of the human genome and the chimpanzee genome that were just too repetitive and convoluted and complicated to actually sequence, and so they weren't. Now, mind you, these regions aren't thought to have contained highly informative sequences of DNA. After all, less than half of our genome is thought to be functional in a conventional sense. Still, without the genome being sequenced telomere to telomere, end to end, I don't really know that you can say that it's complete. That problem is going to be mostly resolved by the end of this video, but for now I want to talk a little bit about some bigger conceptual questions. How do we actually compare genomes between any two organisms, be they of the same species or of different species? And this is where those indels and those SNPs come in. I'm going to explain them now. Mutations come in many flavors. Sequences of DNA can undergo large-scale deletions or small-scale deletions. They can undergo large-scale duplications or small-scale duplications. They can undergo inversion, where the sequence is backward as compared to how it should be, or in the wrong place as compared to where it should be. So when you read comparative genomics papers and they're talking about indels, that's insertions or deletions. Large-scale deletions, like what we talked about in the beginning, or insertions of chunks of DNA where they ostensibly shouldn't be. And SNP stands for single nucleotide polymorphism, and that's a single difference between the two sequences of one base pair. Of the human and chimpanzee differences reported in 2005, approximately 3% came from indels and approximately 1.2% came from single nucleotide polymorphisms. Comparing genomes is kind of like comparing books. So keeping with that analogy, let's do some comparisons with a sentence that is often attributed to Charles Darwin. Whilst man, however well behaved, at best is but a monkey shaved. This sentence has 12 words and 67 characters, counting quotes, spaces, commas, and letters. If I compare it to itself, these two sentences, as represented on your screen, are 100% similar to one another. Same number of words, same number of characters, in the correct order, the whole 9 yards. But let's mutate our second sentence a little bit and see how it impacts our percent similarity, depending on how we calculate it. I've deleted the word well from the second sentence. So now it's exactly 5 characters shorter than the first sentence, a space plus W-E-L-L. How similar are these two sentences now? Well, it depends on how we're comparing them. A program trying to compare two different genomes, or even just sequences of DNA, has to first align them so it can actually run the comparison. And we're going to see a problem right away. Alignment is easy when the two sentences have the same number of characters or words. You just put them right up next to each other. Let's change the word monkey to the word donkey. Alignment is still a piece of cake because the word number and character number is the same, even though the percent similarity has changed. As you can see too, the percent similarity for the word count is way lower now as compared to character count, because donkey is a completely separate word from monkey, but if we're separating everything out into characters, it's just one change of M to D. Which one should we use? Put a pin in that. Let's go back to our comparison between the two sentences where the second sentence has the word well and a space deleted. If we align them by brute word count or by brute character count, we run into problems immediately. In this basic alignment, after the word however, no characters and no words match between the two sentences. The reason is simple. The deletion in the second sentence offset everything. So it seems like there's no similarity, when in reality the similarity is there, it's just slightly shifted in one direction. Luckily, most tools for genomic comparison have the ability to make alignments and take this kind of difference into account. If our alignment software is capable of this, we can get some actual numbers. So let's have our alignment take this into account, as most softwares can. Now that our sequences are properly aligned, what kind of numbers do we see? In word count, the first sentence is 1 word longer than the second, and in character count, the first sentence is 5 characters longer than the second. So sentence 1 is about 92.5% similar to sentence 2 if we're looking at character count, but about 91.6% similar if we're looking at word count. But sentence 2 is 100% similar to sentence 1 because it can be found in its entirety within it, whether we're looking at characters or words. So how similar are these two sentences? What if instead of deleting the word well from our second sentence, we duplicated it? Assuming we align them in a reasonable way, now the first sentence is 100% identical to the second sentence, but the second sentence has 5 extra characters and 1 extra word. But this is a little bit different from the deletion, isn't it? Because the word well as the characters that make well up are actually found in the first sentence. They're just there twice in the second. Let's pretend that words are genes for a moment and that letters represent base pairs. The gene content is 100% identical between the two, but the sequence identity is a little screwy because 1 word is duplicated in the second sentence. Even though the genes are the same, sentence 2 just has 2 of the well gene, the character number is now 5 characters longer. So how similar are these sentences? Let's get really annoying. What if we just invert well? Same situation where the alignability is 100%, it's easy, but 1 word is inverted. There is a 4-character difference and 1 word difference between the two sentences. But let's pretend that we're calculating similarity on the basis of just character count. Even though the characters are different by 4 full characters between the two sentences, should we count that difference as 4 differences? Or should we count it as 1? The inversion, since it's so easy to spot. Intuitively, I think we want to say, well, it's 4 differences. Count it as 4 differences. But that quickly unravels. Genomes are annoying, and mutations are unpredictable. Sometimes enormous portions of the genomes can change with just a single mutation. What if we inverted the entire second sentence? Are we really going to say that these two things are only 2 to 3% similar to one another, even when we know the specific single mutation that caused such disarray? You can see how quickly this kind of thing can get out of hand if you pay no heed to the types of mutations at play. Now that we understand how the types of mutations can impact alignability and percent similarity, we can look back at that 2005 paper and appreciate those numbers with a little bit more sincerity. Protein coding regions are highly conserved across animals in general, so 99% for a percent similarity between humans and chimps in protein coding regions isn't that surprising, although it is pretty impressive. That 96% similarity number comes from comparing the human and chimpanzee genomes as they were completed at the time while taking into account the types of mutations responsible for the differences that create problems for alignability. As we saw with our sentence example, alignments in genomes can actually take into account insertions and deletions at small and large scale and continue the alignment for comparison. So with indels included, indel is just a portmanteau of insertion and deletion, humans and chimpanzees are about 96% similar across their entire genomes. That 96% number also includes all of those single nucleotide polymorphisms, which make up about 1.23% of the 4% difference between humans and chimpanzees. It's worth noting here, just for the sake of clarity and completeness, that there were fewer insertions and deletions as compared to single nucleotide polymorphisms, but because single nucleotide polymorphisms only impact one base pair, whereas insertions and deletions can impact much larger numbers of individual base pairs, insertions and deletions accounted for more of the overall differences. But alignments, for obvious reasons, have a lot more trouble with inversions and duplications. Using placeholders isn't as easy as it is with insertions and deletions because recognizing a sequence as an inversion or recognizing a sequence as one or multiple duplications is much more difficult for a program to recognize, and humans can't comb through billions of DNA base pairs one at a time. These highly repetitive regions and highly inverted regions are difficult to sequence in the first place. They're extremely difficult to align, and even when the alignment is actually made and we understand what's going on in a comparison, they're difficult to quantify. What if our second sentence had 5 full duplications? Pretending again that each of our words represents a gene, these two organisms share 100% of their genes. It's just our second organism has experienced one or multiple duplication events. But how do we actually compare these things? What's going on? Sentence or organism 2 can only align 20% of the way to sentence or organism 1, even though the only reason it can't align 100% is because it's 5 times longer because of 5 identical duplications. They are 100% identical in gene or word presence, but the count is way off because of the duplications for both word and character. In comparative genomics, those repeats of entire sections are sometimes called paralogs or copy number variations, and they're usually left out of full genomic comparisons that spit out a percent identity between two different species because they're really hard to deal with for reasons we just outlined. Copy number variations are a type of structural difference, a large category of genomic differences between genomes that exist not just at the species level, but at the individual level, making up the bulk of differences between any two individual humans. Put a pin in that concept, it'll be important later. That 96% similarity number takes into account insertions and deletions because it's easy to do, but it doesn't take into account copy number variations and inversions, which make up the bulk of the major size differences between the human and chimpanzee genome. The human genome being about 3.2 billion base pairs and the chimpanzee genome being about 3.8 billion base pairs. Now you might be thinking, that sounds kind of inappropriate, but we're going to come back to that here in a minute. You're probably wondering what the actual percent similarity is if you do take into account copy number variation and inversion, and you treat them as wholesale differences, a nucleotide by nucleotide comparison. Basically, this is the percent similarity you get if you align the smaller genome to the larger genome, taking into account insertions and deletions, as well as everything else, as brute, raw alignability differences. You might already be thinking of some potential issues with this type of comparison, but as I said, put a pin in it for now. Evolutionary biologist and molecular ecologist Richard Buggs took the data from the 2005 paper and calculated this himself in 2008 and came up with a raw alignability of about 70%. While Buggs didn't publish these numbers in the peer-reviewed literature, he did calculate them using data that he got from that 2005 paper, and he's certainly more than qualified to make such a calculation given his background. However, as Buggs later admits on his blog, he did make a couple of unwarranted assumptions about the completeness of the human and chimpanzee genome. Buggs is going to revise this number substantially the more complete the human genome and chimpanzee genome gets, but his conceptual methodology here is going to stay the same, where every difference is counted as a difference when the genomes are aligned. To hammer this home, the 10 base pair long sequence you see in front of you with a duplication of the latter half would come out as being 66% identical. Similarly, the new sequence on your screen, if it were to be inverted, would receive a 0% similarity score due to its 0% alignability. There is a question that can be asked here. Which number is more appropriate when asking the question, how similar are humans and chimpanzees, genetically speaking? Should we report the alignment number, or should we report the number that takes into account the types of mutations? Or should we just report the protein coding number, since the proteins are what do the most important functions? Dr. Buggs would propose that the alignment number is the most important number. But when you look to comparative genomics publications, it seems like typically the field reports the number that takes into account the types of mutations and leaves out things like copy number variation, inversions, and so on and so forth. Mind you, they will report the alignment scores typically in the supplementary information, but it's not included in the percent similarity number that is typically given when you ask the question, how genomically similar are two organisms? For example, in 2013, we got the first comparison of several different members of the family Felidae and saw headlines like this: "Your cat is 95% tiger. But what's actually being measured? According to the paper, cats and tigers are 95% similar when looking at the sequence identity. This seems to be analogous to the 96% for humans and chimps. But if you look at the raw alignment, you'll see that a total of 571 of the 674 tiger scaffolds were aligned to the domestic house cat. So they're 95% similar for the 84% of the genomes which were aligned. So this headline is accurate for the portions of the genome which can be compared. It's just not all of the genome can easily be compared. Now here's the wonderful thing about comparative genomics. You can make a phylogenetic tree, or a species-level family tree, using any of those numbers, as long as you're consistent across the species which are being considered. Fortunately, in comparative genomics, usually where there's a percent similarity of any category, there's also a phylogenetic tree. Let's look at some of the reported percent similarities and reported percent alignments for several pairs of organisms in between 2005 and the paper that we're going to be getting into just a little bit today, the most recent completion of Great Ape Genomes in 2025. Keep in mind, this is not a comprehensive list, and several other organisms had their draft genomes reported during this time period, so this isn't all there is. Large portions of the closest living relative of common chimpanzees, the bonobos, genome was sequenced in 2013 by Prüfer and colleagues. Similar to the chimpanzee, the aligned regions reported around 99% similarity to humans. From my understanding, this is the percent similarity after the two genomes were aligned, so it's taking into account single nucleotide polymorphisms, but not indels, copy number variations, or inversions. The alignment number can be calculated by looking at one of the figures, where the chimpanzee contig number is around 3 billion base pairs, with the alignment to the human is about 2.72 billion base pairs, meaning there is about a 90% alignment. Looking at the trees produced in this paper, we see that chimpanzees and bonobos are understandably more closely related than either is to humans. But there's incomplete lineage sorting going on. This was one of the first papers to really flesh out that concept, which I won't get into here, but it's really fascinating and explains why genes are in the places that they are in some of the great ape species. Another paper by Rogers and Gibbs around the same time period produced a wonderful phylogenetic tree that included more than just humans, chimpanzees, and bonobos, that recapitulated the same relationship. In this family tree, again looking at those shared alignable regions and single nucleotide polymorphisms being taken into account, humans, chimpanzees, and bonobos are all more closely related to one another than any is to a gorilla. The 4 of them are more closely related to one another than any is to an orangutan, and the 5 of them are more closely related to one another than any is to a gibbon. This paper likewise mentions that the percent similarity will change if you include indels in addition to single nucleotide polymorphisms, and will change further if you just look at raw alignment numbers. These numbers were roughly recapitulated again by Prado Martinez and colleagues with the same phylogenetic tree. Mind you, each of these papers aren't using the same data set. In most cases, they're sequencing their own genomes and adding dozens of individuals to their analyses. Like, the work of those last 3 papers were going on concurrently with one another. They had to do their own legwork. By now, it's pretty clear that the old phylogeny is wrong. Genetics has shown that humans, chimpanzees, gorillas, and orangutans belong to Hominidae together. Thus, Hominoidea has 2 families, Hominidae and Hylobatidae. Pangenome is no more. In 2018, Cronenberg and colleagues published high-resolution comparative analysis of great ape genomes. These still weren't complete, but they were a significant improvement on prior sequences. Cronenberg and colleagues produced the same phylogenetic tree and same general numbers for protein coding regions and for alignments. Around 99% similarity for looking at the alignable regions of the genome and taking single nucleotide polymorphisms into account, and somewhere in between 80 and 90% when looking at the alignability of any 2 ape genomes. Critically, again, it didn't matter which mode of comparison you were looking at. Humans and chimpanzees, or the humans and the chimps plus the bonobos, were still more similar to one another than any was to, like, gorillas or orangutans. And remember, the genomes still aren't entirely sequenced. So this is where Dr. Richard Buggs comes in again with a new minimum and maximum estimate for those alignability numbers if we assume the remaining portions of the human are maximally similar or maximally dissimilar to chimpanzees. And I really like this blog post because it actually breaks down the portions of dissimilarity into categories, like what portion comes from copy number variations, what portion comes from indels, what portion comes from the regions just being unalignable, and we don't know if it's going to hash out as being more or less similar in the end. You might be wondering at this point, why am I mentioning Richard Buggs' blog posts so much if they're not a part of the published peer-reviewed literature? There is a reason, and I'm going to mention it at the end. Still, none of these genomes were actually complete. They had high coverage, but none of them were telomere-to-telomere. As attempts to get the full genome sequenced for humans and other apes continued, new methods for aligning genomes came to the peer-reviewed literature as well. I'm going to mention two of note here because of the statistics they produced. Marseille and colleagues' Moomer-4 method produced the same numbers we've seen before for single nucleotide polymorphism similarity, indels included, and alignment. And we got the same general numbers again from a paper co-authored by Richard Buggs. The numbers are fairly easily found in the main text, and you can also find a pretty cool figure that shows a sequence alignment. In 2020, Suntsova and Busden published a review paper that recapitulates much of what we've already talked about. But the same exact theme reigns supreme across all of these papers, where the percent similarity between humans and chimpanzees in the protein coding regions was over 99%, including single nucleotide polymorphisms, roughly the same, including indels, about 96%, and just the basic alignment treating all those differences as individual differences contributing to the overall percent similarity. The number ranged from 80 to 90%. In 2022, we finally got the very first telomere-to-telomere, absolutely 100% complete human genome. And shortly after that, we got the first complete ape sex chromosome sequenced telomere-to-telomere. So now we could fully compare the X and Y chromosomes, the sex chromosomes, of humans and other great apes. So what did we find? We got the same phylogenetic tree that we've always gotten. We also got a matrix of alignments for the highly similar X chromosome and notoriously divergent Y chromosome. The fact that these telomere-to-telomere assemblies were now out meant that it wasn't long before we got telomere-to-telomere assemblies for the entire genome for these great ape species. And sure enough, in 2025, you and colleagues released the complete genomes for ape species. So I want to go over some of this paper with you. To make sure I understood what I was talking about, I ran both my notes and this video by Linnea Smeds, who is one of the authors indicated here in this yellow box. Let's see what we found out now that we've got these complete ape genomes. As always, we'll begin with the abstract. The abstract begins saying the most dynamic and repetitive regions of great ape genomes have traditionally been excluded from comparative studies. This is what we've been talking about the entire video, right? This is those alignment numbers, right? The numbers that are typically not included because they're difficult to sequence and they're also difficult to align. Consequently, our understanding of the evolution of our species is incomplete. Here we present a haplotype-resolved reference genome and comparative analysis of 6 ape species: the chimp, bonobo, gorilla, Bornean orangutan, Sumatran orangutan, and the Simang. We achieve chromosome-level contiguity and substantial sequence accuracy, and complete sequence with 215 gapless chromosomes, telomere-to-telomere. Wowza! We resolve challenging regions, such as the major histocompatibility complex and immunoglobulin loci, to provide in-depth evolutionary insights. Comparative analyses enabled investigations of the evolution and diversity of regions previously uncharacterized or incompletely studied without bias from mapping to the human reference genome. So again, this is an issue of early on, some of our ape genomes were biased. They were humanized because the human genome was used as sort of like a scaffold or a reference. This paper does not have that problem. Such regions include newly minted gene families and lineage-specific segmental duplications, centromeric DNA, acrocentric chromosomes, and subterminal heterochromatin. This resource serves as a comprehensive baseline for future evolutionary studies of humans and our closest living ape relatives. In the main portion of the text, we get some background. They talk about much of what we've discussed in this video, that the repetitive nature of ape genomes has made complete assemblies super difficult, and that we need these complete genome genome assemblies in order to ask some really important questions and answer some really important questions. So they tell us sort of what they've done here in the ape genome assembly section and give us some statistics for like how complete and accurate these genomes are. The little sentence that I like the most with regard to this is down here where they say, by contrast, comparisons that included hi-fi data suggested even higher accuracy, one error for every 1 million base pairs. Overall, we estimated that 99.2 to 99.9% of each genome was completely and accurately assembled, including heterochromatin. That's absolutely wild. And they say that these values are consistent with the telomere-to-telomere assembly that was published in 2022 for the human. So this is all good. This means that we're going to be looking at apples to apples here, and we're going to get some really cool numbers as a result. The first figure that we see here is chromosome 7 and chromosome 16, with Homo sapiens here on the top, followed by the common chimp, Pan troglodytes, Pan paniscus, the bonobo, gorillas, specifically as gorilla, Pongo pygmaeus, and Pongo abellii. And then underneath, it's the same thing, same order on the side. And these colors here are telling us the direct alignment, inverted alignment, which means that you've got that inversion, so it's in the wrong direction. And then the regions are highlighted here with centromeres, satellites, and subterminal heterochromatin. So what you can see is that by and large, these two chromosomes are like pretty dang similar to one another, but the biggest differences are between the African ape cluster, so gorillas, bonobos, chimps, and humans, and orangutans, which is kind of odd. These chromosomes being that divergent already between the African ape cluster of humans, chimps, bonobos, and gorillas versus the Asian apes, orangutans, and to a lesser extent the siamangs, tells us that we're probably going to see a difference in divergence times as well. I've skipped down to figure 2 so that you can see that, yeah, that's exactly what's going on here. We've got our phylogenetic tree, and the order is the same order that it has always been in, with Pan troglodytes and Pan paniscus being down here, here's common chimps, and here's bonobos, having a divergence date of about 1.6 million years ago. Then there's humans as a part of this triad, and humans diverged from the Pannon lineage roughly 6.2 million years ago. That's pretty close to the conventional number that has been in place for quite some time, of around 7 million years, in alignment with Sahelanthropus chidensis, roughly speaking. Then you have gorillas as splitting from the hominins and the pannons over here, roughly 10.7 million years ago. That's a little bit older, but not like completely out of the question. The big surprise is obviously where orangutans split off from the African ape clade, which has been pushed back to 19.1 million years ago. That's really old. Like, I study Miocene apes that are like around this time. This is early Miocene, like epoch of time. That's a long time ago, and that's a pretty early divergence for members of Pongo from the other hominids. That's odd to me. And the gibbons up here have been pushed back as well to 24.3 million years ago. And originally the divergence date for these guys was like 20 and under million years ago. So that's the surprising part to me for what I study. This is like extremely relevant for my work with Miocene apes, is the divergence of this Asian group of apes over here from the African ape clade. Obviously, this is something that the authors note as well in the section Ape Pangenome, specifically under Divergence and Selection. They talk about how the sequence comparisons amongst the great ape genomes have revealed a greater divergence than previously estimated. Indeed, 12.5 to 27.3% of an ape genome failed to align or was inconsistent with a simple 1-to-1 alignment, thereby introducing gaps. Gap divergence showed a 5-fold to 15-fold difference in the number of affected megabases when compared to single nucleotide variants, which was due to rapidly evolving and structurally variant regions of the genome, as well as technical limitations of alignment in repetitive regions. So this is exactly what we have been talking about the entire time, that what's causing the differences in alignment that we see and why that seems so disparate when compared to the percent identity or percent similarity when you look at what is alignable is due to these massive structural variants in these just super repetitive areas. But what I'll note, and we're going to see this again in a second, is that the differences aren't like evident between humans and the rest of the apes. They're evident within all of the ape species, and this is going to be something that's going to continue to be highlighted in this paper, especially when we look at the orangutans. They talk about the nature of these big structural differences and how they're located in the predictable regions that we've appreciated for quite some time: centromeres, acrocentric short arms, and subterminal heterochromatic caps, but that they also included numerous gene-rich, structurally divergent regions enriched at the breakpoints of large-scale rearrangement. So it's not just what we've always expected. There's a little bit new going on here too that we'll touch on hopefully here in a second. But they get down to the divergent states and they say our analyses dated the human chimpanzee split between 5.5 and 6.3 million years ago, and the African ape split at between 10.6 and 10.9 million years ago. I will note that this, as I said earlier, is not substantially different than the typical model for the divergence of these groups, right? Typically what I give when I'm talking on my YouTube channel and when I'm talking to my students is about 7 million years for humans and chimpanzees, but that's based off of Sahelanthropus chidensis in part and previous molecular clock estimates, and there's always a range there. The real surprise is, as we said, the orangutan split. The orangutan split at 18.2 to 19.6 million years ago. That's almost double the length of the time between us and the split with gorillas, like us and the chimpanzees and our split with gorillas. That's wild. If we look back at figure 2, you can see where some of the monkey wrenches are coming from. In figure 1A, or excuse me, figure 2A here, you can see the lineage-specific SDRs, or structurally divergent regions, for Pongo abellii, that's one of our orangutan species, Pongo pygmaeus, our other orangutan species, our gorillas here, Homo sapiens, Panpaniscus, the bonobos, and Pantroglodytes, or common chimpanzees down here. And you can see the gorillas just have a massive amount of structurally divergent regions compared to everybody else. And humans, interestingly enough, appear to have the smallest amount, which is surprising to me. Right after that figure, we get one of the first of our important numbers, right? So our phylogeny, check, that's recapitulated the same phylogeny that we've seen, since the beginning of all of this stuff. But now we get our protein coding region numbers down here, or like a pretty good indication of our protein coding numbers, as you will see. They say the number of protein coding genes was comparable amongst the different apes, with slightly more than 1,000 genes predicted to be gained or duplicated or lost when compared to humans. Using the University of California Santa Cruz's gene set based on GenCode, we estimated that 99 to 99.6% of the corresponding human genes were now represented amongst the other apes, and over 90% of genes were full length. That was wild to me. I had to check that with Dr. Smeds because I was like, that's an incredible amount of shared protein coding regions amongst the apes, but it is, here we are. The number has gone up for protein coding regions. The way I'd actually phrased it was, so this paper is saying that 99 to 99.6% of human genes are found in the other apes, and her response was, well, I wouldn't call them human genes, right? Because they're shared in other apes, so by virtue of that, they're ape genes, and we share them with them. There's a bit here that's really cool on the genes that they identified which were human-specific, and they're ones that we've talked about in some cases on this channel before. They say we identified human-specific genes associated with the human evolution of the frontal cortex, including SR-GAP2C, RGAP11, and the Notch2NL gene, which is implicated in neuronal intranuclear inclusion disease. That's really cool, right? Like, this is why it's important to sequence the genomes of other apes, because it has bearing on what does actually make humans unique, even though an enormous portion is shared amongst us all. So, we have our numbers now. We know what the percent similarity is looking at those protein coding regions, and we have our phylogeny. What do we get for those alignments? We already got a hint at it earlier. They already mentioned that there's massive portions that can't be aligned amongst many of these different apes because of the complexities of structural rearrangements and repeats. Fortunately, that number is found in detail in the supplementary material. This is a pretty cool table in the supplementary material, and I'm going to pitch it to you as I understand it, since I'm having somebody who's actually an expert watch this before you see it. Hopefully it's correct. Supplementary table 317 is the alignment coverage for the brand new telomere-to-telomere human genome across the African ape pangenome. So pangenome is basically just the word that that we're using for like all of the other genomes for which we can make this comparison within the African ape clade. So it's the telomere-to-telomere brand new human genome compared against the old human reference genome, another human genome that's relatively complete, the chimpanzee, the bonobo, and the gorilla, as well as the X chromosomes for all of those guys and the Y chromosomes for all of those guys, along with some other additional comparisons here for like alternative portions of genomes, if that makes sense. What we can see here across the top is the region. So the autosomes are the non-sex chromosomes. And then, of course, we have our sex chromosomes, the length of the genome, the query, so the name, our alignment percentage, our identical percentage, and then our alignment 1-to-1 percentage and our identical 1-to-1 percentage. I'm going to focus on this identical 1-to-1 percentage over here, since that's typically the number that you see when we're talking about the raw alignment numbers. I believe this is the number that we've been seeing the entire time when we're talking about the raw alignability of the chimp to the human or vice versa. This is what I think is super interesting. The new telomere-to-telomere human genome aligns 92% of the way 1-to-1 with the human reference genome. That is way lower than you would expect two species to have for a percent similarity, isn't it? We get a similar number for our other more complete human genome here, HG002. Pretty high, 93%, but not identical, right? What the heck is going on here? Because these are complete genomes, right? Or in the case of the ape genomes, like very nearly complete. And yet these numbers are not 99.9%, which is the number that I said at the very beginning that humans share with one another, like across the board. So like, what's the deal? I'll tell you in a second. The chimpanzee hashes out at 87.78. That's pretty close to the numbers that we've been seeing the whole time. Likewise, 87.71 for the bonobo. That's pretty similar as we would expect, since chimps and bonobos are really similar to one another, and slightly lower again for the gorilla at 86.14. All in all, not very surprising for our alignment numbers. With like one major exception, right? Why are the human alignments not more similar to one another? Why aren't the 1-to-1 percent identical alignments higher for the human reference genome to the new telomere-to-telomere or to this other human genome, HG002? Well, that's not 99.9%. Well, the reason is because those structural differences, those duplications, those inversions, those repeats, those things accumulate a lot faster than one might intuitively think. Shortly after the telomere-to-telomere human genome dropped, people started investigating differences between that genome and other fairly complete human genomes. This paper from July of 2023 points out that two human genomes might have more than 6% difference between the two of them, more than 6% being non-syntenic. That number is pretty close to the 93-92% similarity reported in the supplementary material for human genome comparisons to other humans in the Great Ape paper. But my friend Joel Duff pointed me towards this paper, a telomere-to-telomere assembled diploid reference genome for Han Chinese, so for the Han Chinese population, that shows an even greater dissimilarity between two completed human genomes than what we've seen so far. In the abstract, we can find a pretty important line. Each haplotype of this new telomere-to-telomere Han Chinese individual genome possesses 330 megabase exclusive sequences, around 3,100 unique genes, and tens of thousands of nucleotide and structural variations as compared to CHM13, highlighting the necessity of a population stratified reference genome. So CHM13, that's this genome right here, the one that we're using as our telomere-to-telomere complete genome in the Great Ape Sequences paper. The haploid human genome is about 3.2 billion base pairs. That means 330 million base pairs represents about a 10% difference between this new Han telomere-to-telomere genome and the current telomere-to-telomere human reference genome, the one from our Grade 8 paper. And it looks like this isn't even necessarily unusual for apes in general, because Supplementary Table 318 compares the Bornean orangutan primary genome to the orangutan pangenome as a whole. So now our reference genome here is the Bornean orangutan, and we're comparing it to a bunch of other guys, including another Bornean orangutan genome, the Sumatran orangutan genome, and the sex chromosomes for a couple of them. And what we get is something similar, a 90% identical alignability to the other Bornean orangutan, slightly less than that for the Sumatran orangutan, which is precisely what we would expect. The reason for this is pretty simple. Big changes can occur in the regions of the genome that don't do anything critical for the organism, even generation to generation, and that leads to large-scale differences in alignment percent similarity between two individuals, even if they're of the same species. It's even crazier than that. As Dr. Smets pointed out to me, for Sumatran orangutans and for gorillas, looking at the chromosome 4's centromeric region, two haplotypes can differ at the megabase level, right? That's two haplotypes within the same individual differing at the megabase level. That's insane. And this is precisely why the percent similarity that we get from alignments aren't typically used when we're trying to run a phylogenetic tree at the species level. They're variable at the individual level. They're subject to rapid amounts of change, evolutionarily speaking. So we turn instead to the more conserved region of the genome, which can be more informative. It's as true for humans and chimps as it is for other pairs of organisms in comparative genomics. The paper's supplementary material reveals that this holds just as true for chimps and bonobos or Sumatran and Bornean orangutans as it does for humans and chimps. But of course, the kicker is, even if you use those alignment numbers, you're still going to get the conventional phylogeny for the organisms in question. Even if we sequence the complete genome telomere to telomere of every human on the planet and every chimp on the planet and every gorilla on the planet, all the humans are going to be most similar to one another. Then the humans and chimps will form a sister group, and then the gorilla will be the odd man out. And you can theoretically do this with the entire tree of life, with every genome on the face of the earth. You can sequence them all and get a phylogeny. This paper goes over the newly characterized regions that they're responsible for to make these complete telomere to telomere ape genomes. They talk about the areas that still need a little bit more work, and they talk about some of the highlights for what's going on when we look at the great apes and really the apes in general, so the hominids versus the hominoids. They say, perhaps most notably, we provide an evolutionary framework for understanding the about 10 to 15% of highly divergent, previously inaccessible regions of ape genomes. In this regard, we highlight a few noteworthy findings. They talk about how orangutans stand out in showing the greatest amount of SDs at the genus level, but that African apes as a whole still stand out as having the largest number overall. I hope I'm understanding that correctly. They talk about the large-scale differences that are found across the clade in acrocentric chromosomes, the lineage-specific gene families, expansions, explosions, and rearrangements, the weird nature of bonobos with these teeny tiny centromeres as compared to their closest relatives, the common chimpanzees, who have much larger centromeres. They talk about the estimated divergence of the pan lineage. Such 300-fold reductions in size must have occurred recently, perhaps in the last million years. That's wild that that much change can happen just in between bonobos and chimpanzees, just within the last million years. They finish off by talking a little bit about where they need to go from here, the areas that still need to have a little bit more sequencing done, the fact that annotations are still human biased, and we probably need to pay closer attention to gene annotations of non-human apes. It's really a phenomenal paper, and it truly does set the baseline for comparative genomics amongst the hominoids moving forward. But this paper illustrates better than any other paper that has been released so far, the importance of clarifying what it actually is that you're comparing when you ask for a percent similarity between any two species. Especially because, as this paper revealed, those large-scale differences between alignments, that difference in alignment percent similarity, can be major, not just at the species level, but at the population level or even at the individual level. And that's perhaps a good reason for not using it as the answer to the question, how similar are two organisms, genomically speaking? Those highly disparate regions don't seem to occur in the protein coding or regulatory portions of the genome, so they're not subject to selection. That's what lets them accumulate so rapidly. At least for the most part. How similar are humans and chimpanzees? Well, it depends on what you're measuring. In protein coding regions, we're over 99% similar. If you take into account single nucleotide polymorphisms, it's still around 99%. If you include indels, it's about 96%. And if you just look at raw alignability, it drops to about 86%. Likewise, humans and other humans are over 99% similar looking at the protein coding regions, about the same amount if you include single nucleotide polymorphisms, less than that if we include indels, and only about 90 to 94% similar to one another if you look at raw alignability numbers. The similarity of two organisms looking at the protein coding regions, or SNPs included or indels included, is always going to be higher than the percent similarity if you're just looking at raw alignment scores. That goes for every organism that I can think of. The important part is, humans will always be more similar to other humans than any human on the planet is to a chimpanzee. Likewise, humans and chimps will always be more similar to one another by every method of comparison than either of us is to a gorilla. Humans and chimpanzees are more similar, no matter the method of comparison, than tigers are to domestic cats, than Asian elephants are to African elephants, and than rats are to mice. It can be hard to put a specific number to what percent chimp you are, but you're 100% ape. 100% hominoid. There are still so many questions about hominoid diversity and evolution, but fortunately this phenomenal new paper has provided us with a great way forward. Boy, I sure hope no one out there is misrepresenting its results, or perhaps being deceitful about them. That would be really bad. That would be something that I would have to look into. So, my gentle and of course very modern apes, if you like what I do here on this channel, please consider supporting me in the free way by liking, commenting, and subscribing. And if you really enjoy what I do and want to help me out in a financial way, you can join my Patreon. Patrons get their names at the ends of videos, and on the very rare occasion that I'm ahead of schedule, they get early access to videos. And so, from the bottom of my gentle and of course very modern heart, I want to thank you so much for being a curious primate, and I'll see you real soon next time.